Thank you very much for the organizers for organizing really one of the most interesting conferences I've been to in a very long time. There's been a number of talks that have blown me away. And we've heard a lot of talks, or we've heard several really great presentations about using phage as therapeutics and taking advantage of their antibiotic properties. And in the previous talk, it nicely set up what I want to talk to you about. It talked, in the previous talk, there was this wonderful reference to using phage as devices and as nanocarriers. And I want to talk to you today about using phage as the integral elements in electronic devices, using them as the sensing elements that we're going to be using for detecting cancer biomarkers. Okay. Okay. What am I doing wrong here?
Oh, you're using the wrong one.
Oh, of course. Okay, well that makes a lot of sense. Thank you. All right. I'm also going to work in reverse order. So I'm going to start I'll start with a quick disclosure. So the work I'm talking about today has been licensed to a local biotech company that actually just kicked off, got started in December of last year of 2014 and is opening in the next month or so in incubator space next to UCI's campus. I'm a co-founder along with my colleague Reg Penner and Rich Henson as the CEO. Okay, so all of the work I'm telling you about is a collaboration with Reg Penner and his laboratory. This is the Penner team. Specifically, I'm going to be talking about work that's being done by Kristen Eggers, and previously Keith Donovan was a former student who's graduated from his laboratory. In my laboratory, I'm really lucky to work with an extraordinary team of talented individuals, including Krithika Mohan, who did all the work I'm going to be talking to you about today. She's assisted in this by a tiny army of undergraduates, Sasan Zamaradian, Stephen Kudlasik, and Rebecca Drake, who was a summer student in the laboratory. Stephen Kudlasik in particular is just on fire in the lab. Okay, so the inspiration for this, the inspiration of using viruses as nanometer scale devices comes from the great Richard Feynman, one of those really, truly extraordinarily visionary thinkers in the 20th century. And in a clarion call that he made at a physical society meeting, Richard Feynman said, there's plenty of room at the bottom, meaning there's plenty of room to build infinitesimally small devices that would have interesting properties. And this is inspirational because as we start driving down to devices that are similar in size to the molecules found in biology, we're going to have interesting physical properties, such as very high surface to volume ratios that will provide exceptionally high sensitivity. Now, the viruses that we've been talking about at this meeting set us up for that future, because the viruses that we've been talking about, the phage that we've been discussing, actually are, in fact, nanoparticles. And they are on the same scale of nanometers to microns. And an example of the types of things that are coming out in the future is an example from Angela Belcher's laboratory at MIT, shown over here, where she used phage as templates to build lithium-ion batteries. And these have some really interesting properties that otherwise would be inaccessible unless you get down to this nanometer scale. The other thing I like about bacteriophage as tools in our toolbox is that they're relatively easy to manipulate using genetics and genetic control. So we can program in different structures on their surfaces using all of the tools of molecular biology. The other thing is they're inexpensive to synthesize. We can do evolution with them, and I'll show you that in a moment. And they're also extraordinarily stable. The M13 bacteriophage that I'm going to show you in a moment, we routinely pasteurize by heating up solutions containing it to 90 degrees and even 95 degrees and holding it there for a few minutes, and they're fine. They shrug and go about their viral business. They're also stable over a broad range of pHs and in different chemical conditions that most proteins would not tolerate. They're also stable to being dried out. Okay, so if we're going to build devices that are going to detect, in this case, cancer, there are 4 challenges that we have to meet. The first of these is that we need to identify what biomarkers we wish to target. And then the second part is we need phages that will recognize those target biomarkers and ignore other substances. Then we need some way of transducing the act of grabbing onto those biomarkers and turning it into an electrical signal that can then be amplified. And so I'll take each one of those 4 today in turn. So the cancer, the virus, I'll show you virus plastic in a moment, and then finally we'll end with disease detection. Okay, so 4 little pictures that you'll see. So in terms of cancer, I'll start with bladder cancer, and the reason is, first, bladder cancer is diagnosed too late, and when it is diagnosed late, where 54% of cases are diagnosed in stage 3 or 4, the prognosis is poor. This is relative 5-year survival, and you can see the prognosis drops off dramatically. If we were better at diagnosing the cancer early, we'd be more successful at treating it. Furthermore, this is a cancer that comes back 50% of the time after patients are treated. And so the standard of care is to put the patients through having a cytoscope put up their urethra every 3 months to image the bladder and see whether or not the cancer is returned. We can do better than this. We have to do better than this. Cancer is a molecular disease. The molecules that cause cancer are driving cells to run amok, and those molecules are appearing in the bladder, they're appearing in the urine. And so if we learn to detect those and we develop devices that can detect those, we can start changing these grim statistics over here. So that's what I want to do. So the first step is identifying biomarkers. My laboratory does not work on that. Instead, we talk to clinicians. We read papers like this one for examples of biomarkers that we should detect. And then we set up arrays of these we set up arrays of phage that recognize these biomarkers to decipher which ones are going to be potentially most useful. Okay, so this is what we're driving towards. We're driving towards something that would take a urine sample over here and then in a probe manner, dip in and then read out on different channels or in a microarray format different diseases that have biomarkers showing up in the urine. Okay, so in order to make this happen, we're going to need lots and lots of receptors, lots and lots of phage. Fortunately, as you know, phage are an abundant resource. The other thing is we need to make our phage specific to cancer biomarkers, and then we need some sensitive, power-efficient, and inexpensive transducer. All right, let's talk very briefly about the phage. This is an AFM image of phage on mica taken by Teresa McIntyre. So I love phage because unlike the conventional source of binding proteins used for detection, such as antibodies, phage have the stability that I already discussed with you. They're okay in some organic solvents, not all. They're certainly fine in detergents. Their affinities are in the same range as antibodies in this left column. And the specificity is tailorizable and programmable using evolution, whereas it's a lot less specifiable in antibodies. The other thing is that at the end of the day, these guys are easy to make and inexpensive to make because you grow them up in E. coli. Okay, so the phage I'm going to be talking about today, we're going to tailor using the techniques of phage display, and we're going to be specifically targeting the major coat protein of this virus. So this coat protein is being altered using a phagemid, single-stranded, closed circular piece of DNA that's encapsulated by this virus. Okay, this is not a novel idea. There's a whole bunch of other laboratories that have looked at using phage in biosensors. The different geometries are shown here. The different examples are shown in this table over here. Batteries are going on this one. Would it be possible to get more batteries?
There is so you can use that.
Oh, I can use that. Okay, great. All right, and then there's a number of different architectures. We've explored these different architectures where you try to make monolayers of phage. And I can tell you in advance, it's a very idiosyncratic exercise. So I want to share with you today a much more effective way to build devices based on phage. Okay, so I told you that we also need phage that will bind to target cancer biomarkers. The way we do this is that we attach the cancer biomarker to some solid support. And again, using this phage display technique, throw a vast diverse library of phage at this cancer biomarker, wash away all the non-binders, and then amplify up the binders in the host for this bacteriophage, which is E. coli. This is an idea that was invented in 1985 by George Smith while he was on sabbatical from the University of Missouri. Okay, so in practice this works well. We can identify phage that have proteins displayed on their surface that bind with high affinity to arbitrary target biomarkers. We're successful at this 60 to 90% of the time, depending on the quality of the target biomarker. Okay, so let's now switch gears. I want to talk to you about how we're putting the viruses directly into electronics. We're actually putting them in, taking advantage of the fact that viruses like these bacteriophage are negatively charged. I'll explain in a moment. Okay, so this is what things are going to look like in a moment. All right, so it's pretty straightforward to make a highly positively charged electrically conductive polymer using an oxidative reaction. So this is an example of this. This polymer is a thiophene-based polymer that's called P-dot. Okay? And then notice that this has a positive charge associated with it and some counterions. So if you do the same electropolymerization, the same oxidative polymerization in the presence of M13 bacteriophage, what happens is the phage get entangled with this polymer. So now you end up with a hybrid material of viruses and electrically conductive organic material. So you can then send electricity through the polymer, and the electricity sort of ignores the viruses that are along for the ride. Okay, so I'll show you shortly what this surface looks like and where those negative charges are. But for now, it makes sense that the viruses are going to have negatively charged surfaces. That forces them to repel away from each other, which in turn makes them more successful at finding new hosts. And in fact, that's a general property of, to my knowledge, all viruses. But I'd love to hear someone tell me I'm wrong. All right, so here's what the material looks like. This is plastic by itself. This is the electrically conductive polymer called P-dot, and the surface is relatively smooth over here, as shown by electron microscopy. When we add our viruses in at low, medium, or high concentrations, we see these fibrils on the surface of the polymer, and these fibrils correspond to knotty tangles of the viruses that emerge in this high concentration. And we can shape this material into arbitrary shapes based upon electrodeposition using tricks from photolithography. So it's the same sort of tricks that are used to build the laptops and cell phones that I see around the room. So same sort, we borrow from those ideas. My students put on a bunny suit and go into clean room. And then in this case, we built devices that were based on nanometer scale wires. And you can see these little hairy things going through there. Those are actually the phage entangled in the wire over here. So in practice, we do things at sort of a crude level because we're in experimental mode. So this, for example, used my student Jessica Arter's nail polish. And so this is nail polish over here. Here's the little metal particles embedded in our nail polish. That's a wonderful insulator, by the way. So this nail polish is a great insulator. And then here's our virus P-dot nanowires going through here. Now, because these viruses are endowed with the property of binding to cancer biomarker, suddenly this nanowire also wants to bind to cancer biomarker and will be specific for binding to cancer biomarker, as I'll show you in a moment. And that will change the electrical properties of that nanowire. Okay, so let me show you that on the next slide. Okay, so we're now over here where we're putting everything together and starting to use this to detect biomarkers. All right, so in the examples I'm going to show you, we've done a lot of different examples, some that have phage bridging little gaps, some that have phage and nanometer scale wires, and some that have phage in films. And I'm going to just show you films because it's simple to explain in a short amount of time. Okay, so in this experiment, we're looking at two properties of alternating current. Alternating current has both a capacitance associated with it and also a resistance. And these two properties can be plotted on the same graph in something called the Nyquist plot. So this is 1 over capacitance on the y-axis and resistance in ohms on the x-axis. Over here in red and black, these are controls. One of these controls is just run buffer. Another control is an antibody that does not bind to the phage. Okay, so it's a negative antibody. And notice that the black dots and the red dots match up very nicely with each other. In green, this is the signal that we get when we add the antibody that's known to bind to our viruses. And I'm starting with antibodies, but we'll get to cancer biomarkers in a moment. So, oh, I didn't make note of this, but this is, these are signals at different frequencies of alternating current. So really what we need to do is we need to compare the signal at 1 kilohertz for the positive, the experimental antigen binding, versus 1 kilohertz over here for the control. So our signal is not the little tiny shifts over here. Instead, our signal is the difference between this guy up here and this guy over here. That's what we're comparing. So we get massive signals back out. Okay, how does this work? what's happening is the phage that are embedded into this plastic electrically conductive material contributes to impedance in two forms. Okay, so the impedance here is being measured as the electrons that are flowing down to the underlying substrate, in this case gold. In addition, as ions flow down to the underlying substance, the phage have an opportunity to interfere with them. So when cancer biomarkers over here are binding to the phage, that ends up blocking the ionic transport, the non-coulombic electricity flowing to the electrode over here. Okay, so basically what's happening is electrical transport is being reduced as the ions have a harder time making it through this surface. Okay, now in practice, I can tell you when you do this, phage, as you know, are sticky little beasts. They like to bind to all kinds of things, which is both good and bad. The good part is it's easy to engineer them to bind to other things that you want them to bind to. The bad news is they will also find off-target binding as well. So in practice, what we do is we coat our phage with other binding partners that block such non-specific binding. And so here we have binders, one that's genetically encoded and one that's chemically synthesized, coating the phage and blocking that non-specific binding. And it's nice if that chemically synthesized one contributes to the binding. And then this is my last data slide. This shows a detection of a prostate-specific cancer biomarker in urine in a clinically useful range using the scheme that I showed on the previous slide. So we can detect down to 100 picomolar PSMA in urine. And urine is not a trivial solvent, a trivial buffer to deal with because it's high salt, it's highly variable, it's filled with all kinds of other stuff. And so this is basically what we're using as a start for a future where we start sensing biomarkers at an inexpensive level. Okay, so in conclusion, what I've been showing you is that phage are not just friends to the oceans and Arctic soils of the world, but they're also tremendously useful potentially as nanometer scale devices. And they're exciting because you can engineer them using evolution, the same process that they always go through, to bind to essentially any pathogen or disease biomarker. And I showed you this virus PDOT material. I showed you how it responds to antigen binding. And then finally, how we're putting things together both chemically and biologically. Okay, let me stop here. Thank you very much again.